Hello, students. So today we are going to be talking about cell cycle. I highly suggest that each of you take lots of notes. I would suggest making flashcards, maybe using a Quizlet. You could even use somebody else's Quizlet to help you study these. Drawing is another great way to better understand a lot of the things that I'll be talking about. Please make sure you guys watch the entire video the entire way through. If you guys have any additional questions, please let me know. Make sure you guys take notes of those questions so that we can come to class and discuss those things. Feel free to email me or let me know any additional things that you would like help on, and we're going to go ahead and get started. So like I said, today we're going to be talking about the cell cycle. Now, a lot of us may just think mitosis when we hear cell cycle. And while mitosis is a part of it, it is not the entire thing. So this is the division process that is an integral part of life. This is what's going to allow us for not only have reproduction of cells, but growth and tissue repair as well. The cell cycle is the life of a cell from its formation until its division or until it divides. Remember, you guys can pause as needed to take notes. So let's talk about the organization of DNA. Cells must organize and package their DNA before division. DNA associates with and wraps around proteins known as histones to form nucleosomes. These strings of nucleosomes are then going to form chromatin. Chromatin is the type of DNA that we're going to have in a non-condensed form when the cell is not actively dividing. After DNA replication, though, chromatin is going to condense to form a chromosome. So this dense packing of the DNA is going to allow for an easier cell division. Since DNA was replicated, each of those chromosomes is now going to have a duplicated copy. These copies are going to join together to cause something called sister chromatids. This is exactly the image that you are looking at to your left. Part of those sister chromatids are two important things that we're now going to discuss. One is a centromere. This is the region on each sister chromatid where they are closely attached or most closely attached. The kinetochore are proteins attached to the centromere that link each sister chromatid together to the mitotic spindle. Here we can see it pictured with exactly what I was talking about. Our DNA, which is inside the nucleus, is going to wrap around nucleosomes and then further wrap to make what we call chromatin. When it is time for cell division, they are going to extremely densely compact and to form what we know as chromosomes. Now let's talk about the genome. The genome is all of a cell's genetic information, or DNA. This is different for prokaryotes versus eukaryotes, though. In prokaryotes, we have a single circular DNA. In eukaryotes, we have one or more linear chromosomes, and every single eukaryote has a specific number of chromosomes to that, let's say, species. So humans, while we have 46, and examples chimpanzees have 48. And on a different scale, elephants have 56. So this number is going to vary based on the species or animal that we're talking about. I shouldn't say it on a species level. It's going to vary with each animal that we're talking about for a fact. There can be a lot of similarities within and across certain species or subspecies, so we're not going to go there. So now let's talk about the genome. So there's something called a homologous chromosome or homologous chromosomes. These are two chromosomes, one from mom and one from dad, that are the same length and have the same centromere position. These are going to carry genes controlling the same characteristics. So we can think about this and say, hey, this kind of makes sense because we tend to get some traits from mom and some traits from dad, or have some kind of blending of the traits. This is part of that reason. So this is a practice-free response question that we will be doing in class, but feel free to attempt to do it right now. So now let's talk about the types of cells. We have two different types of cells that we're going to talk about right now. We have somatic cells and gametes. So what are somatic cells? Somatic cells are our body cells. They are diploid, or 2N, meaning they have two sets of chromosomes and one set from each parent. These are going to divide via mitosis. An example of understanding this is with humans. So I previously said that humans have 46 chromosomes. That means 2N equals 46. We now know that there are 23 chromosomes from mom and 23 chromosomes from dad. We then have our gametes. These are our reproductive cells, such as egg and sperm cells. These are haploid, or 1N, or you could just say N, meaning that this is one set of chromosomes. These chromosomes divide via meiosis. Please notice that there is a difference between mitosis and meiosis if you have not learned about it previously. But this unit, we are going to be focusing on mitosis. Humans, when it comes to their gametes, N is equal to 23. Again, this makes sense. We're getting a sperm cell is coming together from dad with an egg cell from mom, meaning 23 from each, which would make sense. Once those combine to form our zygote and begin reproducing, we would have 46 chromosomes once they come together, and that is exactly why I'm saying that. Okay, so this is an overall image of the cell cycle. We are now going to be talking about each of the phases of the cell cycle and how they alternate. So the cell cycle consists of alternating phases of interphase and mitosis. So we start with G1, or growth 1, S phase, G2, mitosis, and then cytokinesis. G1 and G2, or growth 1 and growth 2, are both interphase. So what is interphase? Interphase is the longest portion of the cell cycle, making up approximately 90% of the cell cycle. G1 is the first gap phase. So this is where the cell is going to grow and carry out its normal functions, but it's not dividing as of this point. It's just growing. Our next phase is going to be our S phase, or our synthesis phase. This is where DNA replication and chromosome duplication occurs. Finally, or our next phase, I should say, is going to be our G2, or our second gap phase, which is the final growth and preparation for mitosis. When we are not in interphase, we are in M phase, or going through mitosis. So mitosis is when the nucleus divides. Cytokinesis is when the cytoplasm divides. And mitosis is going to result in two identical diploid daughter cells. We are now going to break down the phases of mitosis. So mitosis is broken down into 5 stages, at least 5 stages that we are going to talk about. 1, prophase. 2, prometaphase. 3, anaphase. 4, telophase. And 4 anaphase, and 5 telophase and cytokinesis. So let's start with our first phase, prophase. There are a few key events that are happening here that are really important for us to discuss and make sure we know. 1, chromatin is going to condense. So what are we going to have now? Chromosomes. Our nucleoli are going to disappear. duplicated chromosomes are now going to appear as sister chromatids, and our mitotic spindle is going to begin to form. In addition, our centrosomes are going to be moving away from each other. In prometaphase, we have two key events going on. One is our nuclear envelope is going to fragment. Two, microtubules are going to enter around the nuclear area and attach to kinetochores, or some are going to attach to kinetochores. So our microtubules are going to allow us to actually have the cell divide. It's really important for the process of mitosis. If we have an issue with these, we're going to have an issue with the cell dividing. This is what's helping our cell pull apart. It's what's helping everything divide and go to separate sides or to opposite poles. Metaphase is our next event. Our key events here are going to include centrosomes are at opposite poles. Chromosomes line up at the metaphase plate. So this is across around the center of the cell. Microtubules are attached to each kinetochore. This means that when it's time for us to split up the chromosomes that we have so that we have two daughter cells, each with 2N, these microtubules are going to help with that. Anaphase is our next event. And the key events in anaphase are going to be our sister chromatin separating and moving to opposite ends of the cell due to the microtubule shortening. This is exactly what I was talking about. The cell is now going to elongate. Our cell is starting to stretch. It is getting ready for it to divide into two separate cells instead of one cell. Again, reminder, our end goal of mitosis is two diploid cells, two identical diploid cells. Our last phase is going to be telophase and cytokinesis. So in telophase and cytokinesis, we have a few key events. One, two daughter nuclei are going to form. The nucleoli are going to reappear and chromosomes are going to become less dense or less condensed. My favorite way for remembering cytokinesis, as silly as this is going to sound, is I was always told a thought of cytokinesis. When thinking of cytokinesis, think of boobs. There's a reason why my high school biology teacher told me the same thing. When this is occurring, we have a cleavage furrow which is going to occur, and we see this contractile ring which is trying to separate and pull apart the two cells. Sorry guys, had to pause for a second. Okay, so I say that because the combination of the cleavage furrow and the way that the two cells look together, the easiest way to recognize cytokinesis is by thinking about it that it looks kind of like two pairs of boobs, or a pair of boobs. Sorry to any parents who hear that, but I promise you it is the easiest and best way to remember when cytokinesis is occurring. All right, so cytokinesis occurs in animals when a cleavage furrow appears due to the contractile ring of actin filaments. So this is that kind of like pinching point where we can really divide and separate these, what's going to be two cells. In plants, vesicles produced by the Golgi travel to the middle of a cell and form a cell plate. So now we're able to track maternal and paternal chromosomes because of the way we know it's dividing. We are starting out with a parent cell, which is 2N, or 2N equals 2 chromosomes. After S phase, we are now going to have 2 chromosomes and 4 chromatids. When we enter anaphase, we are going to have 4 chromosomes. Once our cell is going to be able to divide, going through anaphase, going through telophase, going through cytokinesis until it finally is 2 separate diploid cells, each cell is going to be 2N equals 2 and have 2 chromosomes. So we are going to start with our parent chromosome and end up with we're going to start with our parent cell and we are going to end up with 2 daughter cells, each with the same amount of chromosomes as the parent cell originally had. This next slide is going to show us how many chromosomes versus chromatids we are going to have in each phase. So as we can see, in prophase, we're going to have 46 chromosomes and 92 chromatids. In metaphase, 46 and 92 again. By the time we get to anaphase, we're going to have 92 chromosomes and 92 chromatids. In telophase, we're also going to have 92 chromosomes and 92 chromatids. But what's going to happen is when our cell now divides, we are going to cut that number in half and have the exact number of chromosomes that we are supposed to have as human beings. Can you identify the stages of mitosis below? If you think you can, feel free to pause, look at it, and just take a guess. We will be doing an activity or a lab in class where we will be identifying the stages of mitosis ourselves. Okay. Movement of the chromosomes during anaphase would most be affected by a drug that prevents what? Is it A, a nuclear envelope breakdown, B, cell wall formation, C, elongation of microtubules, D, shortening of microtubules, or E, formation of a cleavage furrow? Feel free to pause for a second and go back to your notes and think about what you think the answer would be. Okay, so first let's talk about what's happening during anaphase. During anaphase, our sister chromatids separate and move to opposite ends of the cell due to microtubule shortening. That means if during this phase we're having a shortening of the microtubules, if we are going to affect the chromosomes in this case, or if we're going to affect the movement, a drug that prevents the shortening of microtubules is what's going to most affect anaphase and what would most prevent what's happening. So if we don't want the movement of the chromosomes during anaphase, we'd have to prevent the shortening of microtubules. Okay. Eukaryotic chromatin is composed of A, condensed DNA, B, DNA and RNA, C, DNA and carbohydrates, D, DNA and proteins, or E, DNA and ribosomes. Feel free to take a second. If you said D, you are correct. We talked about the chromatin wrapping with histones. Histones are proteins. Okay, for our next practice multiple choice question, if a sperm cell contains 12 chromosomes, it comes from an animal that has blank chromosomes. So I'm going to turn on my camera and we are going to talk about this for a second. All right. So a sperm cell is a gamete. We said that gametes are 1N or N chromosomes. That means, oh, 1 second, I realize it's a little dark. Okay, hopefully you guys can see. So it's 1N. That means this is going to be half of the total number of chromosomes that the actual animal would have. So we're going to know 2 times 12 is equal to 2N. And we know that N is equal to 12. So that means all we're going to do is multiply. So now we know that 2N is equal to 24. So that means it's going to come from an animal that has 24 chromosomes. Okay, we are going to skip this next practice-free response. We will go back to doing that at a later time. And now we're going to talk about the regulation of the cell cycle. So throughout the cell cycle, there are there are checkpoints. Literally, there are checkpoints. And these are control points that regulate the cell cycle. Cells receive stop and go signals to allow them to know if they should proceed throughout the cell cycle. This is important. We do not want cells continuing to replicate and divide if they have issues or mistakes in them. So major checkpoints. We have our G1 checkpoint. This is the most important checkpoint. This is going to check for cell size, growth factors, and DNA damage. If the DNA is messed up, we either want to repair it or we want the cell to go through apoptosis so that it no longer replicates that DNA. So it's going to have stop and go signals. When it says go, the cell is going to complete the entire cell cycle. If there's something wrong, the cell is going to stop dividing, or it's going to enter a non-dividing state known as G0. So what's G0? This is when some cells stay in G0 forever, or some cells stay in G0 forever, such as muscle and nerve cells. Some cells can be called back into the cell cycle. So if issues that they previously had with DNA or growth are able to be fixed, they can enter back into the cell cycle so they can continue through and eventually go through mitosis. We then have a G2 checkpoint. This checkpoint is going to check for the completion of DNA replication and again, DNA damage. Notice that this is a really important thing that we have to check for. If there are mutations in DNA, we are going to have issues with the cell in general or with the organism. So that means we need to repair these if possible. So if we get a go during this checkpoint, the cell is going to proceed to mitosis. If we get a stop, and if we know that there's an issue with DNA replication, or if we know that there's a DNA damage, if we cannot repair this, then the cell is going to undergo apoptosis. Now, there are ways to repair cell damage, and our cells are constantly trying to do this and going back and basically double checking or proofreading so that they know that their DNA is being accurately replicated. In cases where it cannot fix it, the cell will undergo apoptosis, which is a programmed cell death. Our next checkpoint is going to be our M, or our spindle checkpoint. So this is when we're going to check for the microtubule attachment to the chromosomes at the kinetic core at metaphase. Because remember, during metaphase, our chromosomes are going to line up at the cell plate and prepare for division. If the cell gets the go, the cell will proceed to anaphase and complete mitosis. If it gets the stop, the cell will pause mitosis and allow for spindles to finish attaching to the chromosomes. We also have internal cell cycle regulators. So the regulation of the cell cycle involves an internal control system that consists of proteins known as cyclins. Now the concentration of cyclins vary. Cyclins are synthesized and degraded at specific stages of the cell cycle. So they're going to be specific, and the concentrations are going to vary. We also have enzymes known as cyclin-dependent kinases, also known as CDKs. We have talked about kinases before. So remember that kinases are going to be enzymes which are going to help us move phosphate groups. So the concentration is going to remain constant for each phase of the cell cycle for our CDKs. These are only going to be active though when a specific cyclin is present. So each cyclin CDK complex has a specific regulatory effect. So basically what I'm saying is that active CDK complexes phosphorylate target proteins. They have proteins which are being specifically targeted. This is going to help regulate key events in the cell cycle. We can notice looking at the image that there are varying levels of different cyclins at each stage. We said that there are specific regulatory effects and they target specific proteins, meaning at different phases, we're going to have there's going to be different things that it needs to do, or it'll have different proteins that it needs to phosphorylate. We also have external cell cycle regulators. So one of these include growth factors. These are hormones released by the cells that stimulate cell growth. When this happens, we have a signal transduction pathway that is initiated. We talked about the signal transduction pathway, now it comes back. The CDKs are activated, leading to a progression through the cell cycle. We also have something called contact or density inhibition. This is when cell surface receptors recognize contact with other cells. This is also going to initiate a signal transduction pathway that's going to stop the cell cycle in G1 phase. In addition, we can also have anchorage dependence. This is when cells rely on the attachment to other cells or the extracellular matrix to divide. If they are not attached, if they are not anchored onto something, they're not going to divide. So these are great examples of external cell cycle regulators. So while this is not regulating the cell cycle from within the cell, these are additional factors occurring outside of the cell that can help regulate the cell cycle. Now, let's relate this back to cancer, which is 100% an evasion of the cell cycle. Oops. Sorry guys, I had to yell at my dogs for a second. They decided to fight in the middle of my recording. So normal cells become cancerous through DNA mutations. DNA mutations are changes in the DNA. Cancer cells on average have accumulated anywhere between 60 or more mutations on genes that regulate cell growth. This is why we have tumors that form and why they begin to grow and replicate out of control. So let's talk about normal cells versus cancerous cells. So normal cells are going to follow checkpoints. On average, they're going to divide between 20 and 50 times in a culture. So by this, I'm saying like in a petri dish or in a certain type of media. And they're going to go through apoptosis when there are significant errors. So if we have lots of errors in the DNA, we have all of these checkpoints that we're going to go through that's going to cause us to stop and the cell to go through apoptosis if we have issues or if we have significant issues. With cancerous cells, they're not doing this. They're not following their checkpoints. They are dividing indefinitely. Oftentimes, we refer to cancer cells as immortal. And these evade apoptosis due to continuing to divide even with errors. Because they're not following those checkpoints, because we're not able to signal the cell to undergo apoptosis when we have these errors in DNA, when we have these significant mutations, the cell is going to evade apoptosis and continue to divide. So when we have the uncontrollable growth of cancer cells, this leads to what we call a tumor. This is a mass of tissue formed by abnormal cells. There are different types of masses, though, which are caused by abnormal cells. We have something called a benign tumor, which are cells that are abnormal but not considered to be technically cancerous yet. So certain types of skin cancer, like basal cell carcinoma, we consider those typically benign tumor cells. These cells only remain at the tumor site and are unable to spread anywhere else in the body. We also call this metastasis, or the benign tumors are unable to metastasize and spread to places aside from where the tumor is originally located. We also have something called malignant tumors. This is a mass of cancerous cells that lose their anchorage dependency and can leave the tumor site. This means they can now divide wherever they go and can spread throughout the body, whether it's blood, whether it's your bones, whether it's in your lymph nodes. And we call this metastasis. And this is when cells separate from the tumor and spread elsewhere in the body. Typically, this is when we see deaths from cancer or advanced, and this is when we have advanced stages of cancer. Now, it is important to say that because we are talking about cancer, it's also important for me to say how we can prevent cancer. And though cancer can affect everyone, there are definitely ways in which you can minimize your risk of developing cancer. And there's a reason for this. Not smoking. smoking, such as nicotine and other carcinogens within things like cigarettes or vape products, cause mutations in DNA at significantly higher rates. It's also important to eat healthy and drink water because fatty foods and dehydration can affect the functions of cells. Finally, and trust me, there are many more aside from this as well, protecting your skin by using SPF, and by that I just mean sunscreen, because sun is damaging to skin cells and can cause mutations after exposure. This goes for everyone though. There are a lot of misconceptions that having darker skin means you can't get skin cancer. All it really means is that it's typically harder for you to see the mutations. So it's really important for anybody, regardless of very dark skin or fair skinned, to wear sunscreen. Okay. We are not going to do this practice-free response because we will be doing lots of practice-free responses in class. But please make sure that you guys go over everything, take your time, write all of the notes, and I'm not going to give you guys any little quiz thing on this lesson, but make sure you guys come to class with any questions that you guys have, and we will be doing lots of practice on mitosis and the checkpoints so we have a better understanding. Thank you guys.